everyone. I'm Katie Sacra, the Family Education and Engagement Liaison for the Global Foundation for Peroxisomal Disorders. Thank you for joining today's webinar, Introduction to Genetics, part one of a two-part series. I am grateful and excited to have Dr. Eric Rush presenting to our GFPD families and friends again this year. Dr. Eric Rush is the Medical Director, Office of Faculty Development, and a clinical geneticist at Children's Mercy Hospital and an Associate Professor of Pediatrics at the University of Missouri, Kansas City. He is board certified in internal medicine, pediatrics, and clinical genetics. His research interests include extraskeletal manifestations of osteogenesis imperfecta and bone pathology and inborn errors of metabolism. Dr. Rush is also on the GFPD Scientific Advisory Board. His work has contributed to research publications, including the published treatment guidelines for peroxisome biogenesis disorders in the Zellweger spectrum. And low bone mineral density is a common feature of Zellweger spectrum disorders. Before we get started, please know we will be taking time at the end of this presentation to answer any questions, but you can go ahead and submit your questions throughout. You can do this in the box located beneath the webinar player. Also, you can do this through the chat box feature. Dr. Rush, thank you for joining us today. I'm going to now turn it over to you.
Very good. Thank you so much, Katie, for that kind of introduction. I'm really glad to be here today. I was excited to the chance to talk a little bit about sort of an introduction to genetics. And if the title of my talk seems a little odd, I just want to point out that if you look at the course catalog for the University of Missouri, Kansas City, Biology 206 is Introduction to Genetics. And it is a class that I occasionally will do a couple of lectures for every year. And so you're getting a little bit of the flavor of Biology 206, which is an undergraduate level course in Introduction in Genetics. So hopefully we'll have a lot of fun today. I know I'll have a lot of fun today. And hopefully everybody will learn a little something. And please feel free, after I'm done with the lecture, I certainly won't take all of the allotted time for this. And so there should be ample opportunity for some questions. And so let's get started with what we're going to do today. We're going to talk a little bit about some concepts and some terminology. And so one of the things that I want to be very careful to do is to unpack some of the terminology that we do in genetics. You know, when I was an undergrad in genetics, and even when I was a genetics fellow, one of the things that became very clear to me is how important it is that to understand the language that is used for genetics. And it really is a separate language that we use to describe these concepts. And you know, like all languages, like all highly technical jargon, what it's really trying to do is to boil down very complex concepts or aspects of those concepts into a single word or a very short phrase. That's useful, but unfortunately, it can make it impenetrable for patients. And one of the things that we really want to do, and I can certainly speak for my colleagues on the Scientific Advisory Board, is we really want to make all of this with these very complex concepts as easy as possible and to unpack some of the terminology, because the truth is all of you can absolutely understand this. It's just that we have to help, kind of guide you through some of the language. And we're going to talk a little bit about really how information is stored on DNA and what we mean when we're talking about things like nucleotides or molecular testing or things like that. And I'm also going to give just a little bit of historical context and talk about the technology. And some of this will just be fun to kind of understand what the journey's been over the last 20 years. It's certainly been a fun journey for me over the last 20 years. So when we talk about genetics, really what are we saying here? What do we mean by the study of genetics? And so really, the genetics is the study of two things: heredity and variation. And so when it really came up, when we came up with the term genetics, it was talking about hereditary variation of essential characters of an organism. And so there are these two concepts of heredity and variation are actually somewhat in tension with one another, because there's heredity, meaning the tendency of offspring to resemble their parents. But then there's also variation, which is the tendency of the offspring to vary from their parents. And so those were kind of two inextricably linked concepts that were unified by the whole idea of genetics. And the term genetics as a discipline is about 100 years old to describe more the study of heredity rather than the study of variation, even though we understand them both to be important. And so it's from the like almost all of our terms that we use scientifically, even though they're terms in English, they have their Genesis, which is the exact same root word of the Greek geno, which means to give birth. And so genetics, Genesis, genealogy, they all have the same term, the same Greek root word, which is to give birth, which you might not think is interesting, but I think is interesting. So let's talk a little bit about more about some of the genetic terminology. So when we're talking about things like genotype, and a lot of you have probably heard us use words like genotype. What we're really trying to discuss is genetic makeup, or whatever the genetic composition is of a person. Oftentimes we'll talk about genotype when we're only talking about an alteration to one gene. And, for instance, when we're talking in our families, we might be interested in a genotype for a person for a gene like PEX1. That's a very important thing for us to understand. But we can also talk about genotype in a little more global sense for a person's overall makeup, although that's a little tougher to communicate. When we're talking about phenotype, we're talking about aspects of a person that we can see, or that we can somehow deduce through objectively. What does a genotype actually do to a person, if anything? One of the terms that we used that we probably shouldn't use anymore is the term mutation. So mutation is kind of an old term that it literally means change. And any sort of alteration in the genetic code could be considered a mutation if we are using the most specific definition of the word. The problem with the word mutation is it's in many ways a little bit pejorative. Not only the term mutation, but its other derivatives like mutant. Especially if it's ever applied to a human, is a very pejorative term. And so we really don't like to use that anymore. And the other problem that I have with the word mutation is because it has a negative connotation, when we talk about mutation, people often assume that every change or every mutation is disease causing, which is not true. We prefer the term variant. So what does a variant mean? It means a change. The thing I prefer about variant as a term is it doesn't make a judgment about what the variant is doing. We have variants that are benign, that are what we'd say are not responsible for any sort of effects, or another term we can use that is also problematic is normal. Well, what really is normal? And that definition is not really even clear. Or pathogenic, which means that we find a variant that is responsible for the identified effect. A pathogenic variant is what people think of when they're thinking of mutation, even though pathogenic variant is far more descriptive. And pathogenic just means it's something that is disease causing. Well, but there's also variant of uncertain significance, and some of you in the audience may have been exposed to this information of a variant of uncertain significance, which means that we don't know. And this is, and I'll come back to this a couple of times in this, but this is sort of the dirty little secret, if you will, about genetics, is we don't have it all figured out yet. And a lot of you have lived that with yourselves, with your families, that we don't have it all figured out yet. And sometimes we'll find changes that we're not 100% sure are disease causing, or we're not even maybe 90% sure are disease causing, or 90% sure are benign. And so we have to call them a variant of uncertain significance, which is sort of a bookmark, if you will, sort of a stay tuned. And so you've also probably heard the term genetic versus genomic. So what does that really mean? What I find is that oftentimes they're used interchangeably in the lay press. And in fact, there's a trend over the past 2 years to use the term genomic much more frequently than genetic. It's maybe a little more of an exciting term. It seems newer. It seems more a more interesting term. But don't make the same mistake. Not that I think anybody here would. When I think about genetic, I think about specific aspects of a person's genetic makeup. So if you are doing a test for a series of PEX genes, and you find a change in that in one of those PEX genes, that's genetic testing, because you're looking in specific areas, and you're looking in areas where we have known function. Genomic testing in genomics refers to kind of the whole totality of what we know and what we don't know about a person's genetic makeup. So if you're doing what we would call whole exome or whole genome sequencing, where you're kind of just looking at everything, that's what we would consider to be genomic testing. And the reason, and some of the difference is not just the scale of it, but is also that we're looking at areas that we know what happens when they're not functioning correctly, but we're also looking at areas where we don't know what happens when they're functioning correctly. So what are chromosomes? We've talked a little bit about genetic versus genomic, but let's talk about a little bit about genetic material, not just as a metaphor. Let's talk about this genetic material as an actual physical object. And I think this is something that I find that not just patients and families, but my non-genetics colleagues sometimes don't quite appreciate, is that genetic material is a real physical object that you can explore, and in some cases that you can manipulate. And it starts with chromosomes. So these chromosomes are the actual structures that store genetic information. And that genetic information, those chromosomes are constructed of DNA. And we know, and I imagine all of you on the call know that DNA is the molecule of heredity. It is how our genetic information is stored. Every organism has chromosomes that are packaged a little bit differently. And in the case of humans, we have 46 chromosomes, so 23 pairs of chromosomes, which, fun fact, we actually didn't know that we had 46 chromosomes until well into the 20th century. There was a period of time that it was published that humans had 48 chromosomes, which I think is kind of shocking that this happened. And this was really kind of the 1930s, 1940s. It wasn't all that long ago. 22 pairs of these are what we call autosomes, meaning it's the same structure in males versus females. But one pair is what we call, these are sex chromosomes. So most phenotypic males have, and again, phenotype meaning what a person appears like. So most people with male physiology have one X and one Y chromosome. Most females, phenotypic females, have two X chromosomes. Now I say most because there are exceptions, like there are exceptions to literally everything in genetics. So the chromosomes are kind of the basic physical medium, and this is what chromosomes will look like. If we actually we can break apart a cell and we can put special stains on the chromosomes to make them look like this. And so these are pretty nice chromosomes. Somebody did a pretty good job with these. And this is what we call G banding, the chromosomes. And so you'll see these little kind of stripes on here of dark stained and light stained chromosomes. I won't get too much into the weeds of this, but this is kind of one of the tried and true techniques for how we diagnosed some conditions. Obviously not a peroxisomal disorder. We wouldn't be able to diagnose a peroxisomal disorder on a karyotype. But we can diagnose some things that are a result of whole chromosomes that are extra, like Down syndrome, or whole chromosomes that are missing, like Turner syndrome. So we are able to diagnose some of these conditions on karyotype. It just depends on what you're looking for. And part of my job as a clinical geneticist is picking the right tool for the job, depending on what patient comes into the office. So taking this a little bit further from the chromosome, when you start to unwind the chromosome, you will find that there are big loops of DNA that are wrapped around these big proteins, these kind of glob proteins called histones. And you form these very complex structures in sort of coils of coils, if you will. I think about this almost in the same way if you would take an old telephone cord, and I know I'm aging myself by saying telephone cord. Note to our millennials in the audience, we used to have phones with cords that you plugged into a wall. Crazy, I know. But if you take the telephone cord and wrap it around and then wrap it around again, that almost kind of looks a little bit like the structure that we see in the chromatin that goes on to make the chromosomes and ultimately stores DNA. But as you start to unwind and unwind, you get to the structure of the DNA itself. And this is the double helix that you've probably heard talked about. And so what DNA actually is constructed of, what DNA is actually constructed of is a backbone of a type of sugar called deoxyribose and then phosphate. and they go in sequence, and so it forms the backbone. And that backbone provides the structure for the nucleotides, and those nucleotides are those A's, G's, C's, and T's that we think about when we're talking about the sequence of DNA. And it's that code that is unique for each gene that makes a normal product of that gene. So the gene is kind of the blueprint, if you will, and they all have a product. And if there's a change to that product, then that's when we can see problems. And so in order for us to make that product, there is a process that we have to go through. And so you can look at this even a little like an assembly line. And so in order to make a gene product, you have to have an RNA made from the DNA. And so RNA is a different type of a molecule that has some similarities to DNA, except that it's typically only single-stranded instead of double-stranded, and it's a different sugar, and that is in the backbone called ribose instead of deoxyribose. The interesting thing is, even though we think of RNA being a little bit secondary, it probably came first evolutionarily. We don't know that for sure, but it probably came first. So the RNA is actually transcribed from the DNA, and there are 3 phases of this. So basically, you got to start it, you got to make it, and then you got to end the process. And honestly, that's all you guys got to know about transcription. And then what happens is you have that transcript that leaves the nucleus. So it's you've kind of made a copy of the DNA that you wanted, and so now you have the RNA that you want, and then you can go ahead and make your protein, and the protein is the product from the gene. And so what happens is it's almost like a little shuttle that goes along and starts reading the RNA in 3 letters, in these 3-letter codes called codons. And each one of these will correspond to a different amino acid. And so each amino acid is then just hooked on. And so you get a long series of amino acids. And what's also interesting, as you might imagine, you have to have other instructions there. And so you have an instruction for start, which is an initial amino acid methionine, and then you have your sequence of all of these amino acids. And then you also have instructions for stop that says, okay, we're done here. And then all that apparatus falls away and you're left with a protein product. Now, sometimes you have to do other stuff with the protein to make it do what you want to do. And a lot of times these are part of very complex pathways. But for our processes today, the only thing that I think it's important for you to understand is that there is a process to go from DNA to making RNA to making the final protein product. And that whole process is what we call the central dogma of genetics. And there are, like all good dogmas, there's actually a lot of there can be exceptions to it, because not all RNAs that are made actually make proteins. But for our purposes, that's where we want to think about this. And so in this, you can see that there is one of these codons that is coded in green, which is methionine for start. And there are 3 of these codons that are stop. So you might imagine if there are changes to a person's genetic code, you can have substitutions. You can have changes from one amino acid to a different amino acid, but you can also have stops that are introduced. This is what we call a nonsense change. So it's important to know these because they can have some very different mechanisms of disease. So as far as inheritance patterns are concerned, there are really 2 that we need to think about here, and we'll talk about those, and we'll talk about kind of what some of those mean. But when we're thinking about inheritance, we can think about this in terms of recessive, and we can also think of this in terms of dominant. And for our purposes here, we're only looking at this on the autosomes. So these are not the sex chromosomes; these are the autosomes. In which case we see recessive conditions that will result from the 2 parents, from mother and father, both having changes to one of their genes and then passing along that change, again, that variant, to an offspring. And so typically that will result from either less of activity of a normal function, or maybe completely eliminating the normal function of a gene to have a disease. So typically those people that are carriers, meaning they have one copy of that change, will be healthy, will be okay, but they can pass it along to a child. Now, dominant is a little bit different. Again, it's still located on the autosome, but there's only one copy of a gene changed that needs to change in order to change that normal function and have a disease process. So what is this? So we're going to boil this down a little more for the recessive, because that's really, this being GFPD and knowing what we know about Zellweger spectrum disorders, is we're most concerned about autosomal recessive disease. So when both parents are carriers of a disease, the child inheriting that whatever that change is 1 in 4. So we know that, again, that the chances are 1 in 4. The chances are actually higher for those parents to each pass along one working copy and one non-working copy. So in those cases, it's 50% chance that they would have a child who are clinically unaffected but are carriers. We wouldn't think that they would have any health problems, but we know that it can be a future family planning problem for them. And we also know that pass along a working copy of this, and so those will be unaffected children who are not a carrier. So why is it then that most families with ZSD don't have a known family history if the risk is 1 in 4? This is a question I actually get quite regularly, is we've never seen this in the family before. Why not? And the reason is because your long odds are not what occur when the two parents have already decided to have children together. The long odds are for those people to come together in the first place. Because a lot of times, if you have a condition that has an incidence of 1 in 100,000, so pretty rare, you think, well, what is the carrier rate for something like that if it's 1 in 100,000? As it turns out, the carrier rate's about 1 in 150. And so that's actually a lot less rare than a 1 in 100,000, but it's still more rare than the 1 in 4 risk for people who are already together. So the real odds are those two carriers finding themselves in the first place. Now that happens, and it happens because it's a numbers game, and there's almost 8 billion of us, and so invariably we're going to have situations in which a couple where two partners are carriers for the same condition are eventually going to meet. So those eventually will happen by chance, for lack of a better term. So let's take a little bit of time out for terminology, because these are some terms that I know are thrown around in the genetics community, but I want to make sure everybody understands. And so those terms are the terms homozygous, compound heterozygous, and heterozygous. In the case of homozygous, we would see both copies of a gene, both from mom and from dad, have a change, and it's the same change. And so in the case of a patient with a ZSD, we might be thinking of one of a very common PEX1 variant. Okay, whereas compound heterozygous mean that both copies of a gene are altered, but the change itself is different. You change. Whereas if we say heterozygous, it's only one copy of the gene is altered. And in this case, with the term that I've used, I've identified the variant, and I've used the term WT for wild type. Now, wild type is kind of you could argue is controversial in humans, because there's really no such thing as a wild type human, but it's something that we sometimes will use when we want to express that a person has a normal allele, or a normal genetic state at that position, at that place in the gene code. And so carrier is a term that we will sometimes use to describe heterozygous state in a recessive disease. And so carrier itself is a little bit loaded, because it implies that there are no health implications from that. Although not necessarily with ZSD, but we know there are other conditions where we actually have found out that carriers may have mild disease. And so sometimes, so we have to be very careful how we use that term. So now that we've discussed a little bit about the terminology, how do we actually get to a place where we have a genetic test? How do we actually do this? And so what we would use for a person who is being evaluated for a ZSD is not the karyotype that I discussed earlier, it's DNA sequencing. And so what we're looking at doing is we're looking at reading that order of those bases, those A's, C's, G's, and T's in a DNA molecule, and we're looking for changes at that base pair level. And so just as an example, I've given here of a control, meaning a wild type, where has a T in a certain position, whereas the patient has a C in that position. Because that change occurs, that changes the meaning of the code. Now, we don't know from this information quite yet, is this a nonsense, meaning does that change it to a stop? Or is it a missense that changes it to a different amino acid? Or there is a case where we can have a silent change, where because we have some built-in redundancy here, and I'll go back to that real quick, because you'll see sometimes these codons, some of them will have multiple sets of these codons that encode the same amino acid. And interestingly enough, the amino acids that tend to have multiple codons also tend to be pretty common amino acids. So we'll say that they're kind of building blocks that you want a lot of, tend to have more than one of these codons, whereas the ones that may only have one or two tend to be rare, maybe not so commonly used. Interesting, at least to me. But we really started this whole process a long time ago. And I talked just now about DNA sequencing, again, sort of as a paradigm, but we really haven't been doing a lot of this for very long. And so I think some of what we do makes a little more sense if you understand some of the historical context here. And there really was a humble beginning here. And so the chromosome number of man, which 2020, but in the 1950s, this is what was used. The chromosome number of humans was defined as 46 based on this paper that was done. The technology that we actually started using, the kind of our tried and true technology that we started using, was started in the late 70s. And this is what we call chain terminating DNA sequencing, or what we more commonly call Sanger sequencing, after Fred Sanger, the person who came up with it. Well, it took us a while, but we started to get really interested in ourselves. After all, what's more interesting to humans than humans? And so we said, well, let's look at looking at the Human Genome Project. And they started doing this very slowly and laboriously with Sanger sequencing, kind of that tried and true technology that was invented in the 1970s. But it was expensive, and it was time consuming, and it was slow. And so by the time the Human Genome Project turned about 9 years old, we had really only just mapped the first human chromosome. That was chromosome 22, and it's the smallest human chromosome. So we were really way behind in the Human Genome Project. And I think that was something that people kind of looking back don't quite understand is, even though it turned out to be a really big success, that success was by no means assured. And 9 years into a project that ended up lasting 13 years and was intended to last 15 was very much in jeopardy. And during this time, I was an undergraduate at the University of Kansas, and I was working in a molecular genetics lab, and I saved this email, and I still have the hard copy somewhere, of the graduate student that I was working with, and says, Eric, I need you to be as specific as possible about the ROI, the region of interest. So it's basically what is the area of a gene that we're really, really focused on. And the reason is because we pay by the base pair, and it was about $1 a base pair to do sequencing in 1999. That's crazy. And so $13,000 per megabase in 1999. You don't pay $13,000 for a whole genome sequence in 2020. So I mean, this should just kind of give you an idea of how far we've gone in the past 20 years or so. So this is the Sanger sequencing, again, tried and true, very good technology. But again, it's slow, and that makes it expensive. And so what you get out of this technology is you get these little these kind of reports out that are where you see these kind of colored peaks, and you can then read what the sequence is. So again, we still use Sanger sequencing in some cases, but we just don't need it to be used in all cases at this point. And it's really most helpful if you have something that's very little. So if you want to look at one single gene, it's probably okay to look at with the Sanger sequencing. But if you're wanting to look at lots and lots of different areas, then Sanger sequencing is probably not very good. And so I alluded to the fact that we ended up having a successful outcome for the Human Genome Project, and there's a reason why. And the reason why is that we started using a new and better technology. And so those of you who are in the computer or IT industry may be aware of a law called Moore's Law. And in 1965, Gordon Moore, who worked for a very small company called Fairchild Semiconductor that turned into a not so small company called Intel, observed that the number of components, meaning transistors, into an integrated circuit, so it's kind of an early microprocessor, doubled every year, and eventually we said doubled every 2 years, and then also halved in cost. And so what's interesting is genetic technology completely mirrored Moore's law until 2008, and then it rapidly exceeded Moore's law since that time. Why? Why did that happen? And the reason is new technology, what we now call next generation sequencing. And next generation sequencing really had its start in the mid to late 1990s. And the reason why that developed when it did is honestly is because necessity is the mother of invention. We were behind the ball on everything when it came to the Human Genome Project, and so we needed better technology, we needed better tools. And so what happened is we did the first chromosome in 9 years, and then we basically did all the rest in the next 4 years. And so instead of being done in 2005 with the Human Genome Project like intended, we were done by 2003 because these technologies are so much faster. And so what has been the result of that next generation sequencing? What has been the payoff for us with that? And the payoff is instead of picking one gene or two genes like we did in the past, we're able to look at lots of genes. And in some cases, we're actually able to look at all genes. And so what we started to do is, even though we were able to do this, it was really expensive to look at every single base pair of every single gene, and even areas outside of genes. It was very expensive to do that. And so what we started doing around 2008 is we said, okay, what if we just looked at the areas that we know are part of genes that are actually part of the protein part of it, what we call the coding region? What if we just look at that? What would that do for us? And because it turns out that's only about 1.5%. And so we've just cut down on a lot of work for ourselves, and we're just getting into this, and so that's pretty important. And especially because we kind of the estimate was that really most of the changes, the variants, will be in that area, in the coding regions, and so this would seem to be a pretty good deal, and it is. And so we started doing looking at this cost, and so the cost kept going down. And so you can get exomes really cheap now, but what you're not gonna get is you're not gonna get a nice report from a $300 exome. What you're gonna get is a flash drive with a bunch of data on it that you have to go analyze yourself. And so that's not ideal for most purposes, and it's not what we do clinically still. So why would we use an exome now in 2020? What we really would do it is if we have a person who we're evaluating that we think has a genetic disease, but we don't really know what kind, or we don't really have a prediction as far as what type of condition they have. Or if we have an idea, but they've had a really extensive evaluation and we've never found anything. And so we've kind of done our targeted approach and we've never found anything. And one of the things about exome, and it's true about the whole genome too, is it's really important to get both parents involved as well. Because again, there's a lot of variation that we have as humans that's totally normal. It's a normal part of being human. But we can't exactly predict what's normal or what's not normal just looking at the patient. But if we have two healthy parents in front of us, that can a lot of times really help us in saying, well, this is something that's concerning to us, where, nah, this is probably okay. This is probably not something that we need to be super worried about. And that's really useful. So what are the limitations of exome sequencing? Well, it's whole exome is not really whole exome. It's probably, I say 85 to 90% because I created this slide a couple of years ago originally. It's probably in the mid 90s at this point. So, but it's still not 100%. And what we've also found is that changes to the non-coding region, to areas that are not actually part of a gene, can still be important. They can still disrupt things, because what we found is that even though the genes themselves are probably where most of the changes are kept, there's also parts of all of our genomes that are not necessarily part of genes, but were probably important for helping genes function, what we call regulatory regions. They're probably important for telling genes, you should turn on here, you should turn off here, or you should turn on in the liver and not turn off on in the bone. So these are actually in some cases very important for us to know. And we've also figured out is that the genome is really variable. There's a lot of differences amongst us that we probably, most of it's probably not that important clinically. And what the other thing that's important is that there's about a little over 20,000 human genes, but we really only have human disease that's described in a minority of them, and it's more than 3,800 at this point. And there's a question about CRISPR, and I will get to that. I will get to that with after the presentation. But put a pin in that because I don't want to forget that, because that's a very interesting question. So what have we seen as far as the cost here? And so this is something that I think to me is the most impressive, is that the first whole genome sequence that we did was the Human Genome Project, and it was 13 years and cost $2.7 billion, which is a lot of money and a lot of time. The second whole genome sequencing took 3 years and it would cost $100 million. Doing a whole genome sequencing today, you can do it for as little as $600, and if you and I shall say $600 and a 2-week turnaround time are mutually exclusive. You can get a $600 whole genome. You can get a very short turnaround time. You cannot get them at the same time. And so if you do that, your $600 whole genome is going to take you quite a while to get back. But if you want to do a clinical whole genome sequence, we're probably talking about more like 12 weeks, and probably somewhere around $8,000 to $10,000. That's an estimate. I haven't looked up the clinical price sheet recently, but that's probably about right. So it's still an expensive test, but it's a lot less expensive than it used to be. And so I think that kind of underscores some of the benefits to what we've seen recently, and kind of what next generation sequencing has done for us. So with that, I'll end, and I want to take questions. I'll answer the question sent to everybody about CRISPR. So, is there any potential in the future of the possibility for CRISPR gene editing to be able to stop the progress of ZSD in patients? Is it just being researched for certain areas of the body, such as the retina? So, this is a very controversial topic. So this is gene editing in humans. This is a very controversial topic, and at present there is a moratorium on CRISPR research in humans in the U.S. Not every country has been abided by that, and so you might have read in the popular literature that there's been some controversy over gene edited human embryos that were created, and I think that they're right for having the discussion. So regardless of what you think about the kind of the ethics of gene editing, it's very clear to me that prior to any active discussion of human gene editing clinical trials or research into doing that, we need to be very careful about about the ethical, legal, and social implications for that sort of testing, or that sort of gene editing. And we will have to come up with some very strict guidelines for what that means. Now, I don't know what the outset of that's going to be. Now, we could have those discussions and come up with very restrictive guidelines, up to and including, we don't do this sort of gene editing on humans. That's possible. Maybe that's not written in stone forever, but it's possible. but it could also include something that's a little more permissive to say we support that for these sorts of conditions, but not for others. And I think that it becomes, the concern is not just the gene editing itself, but the concern is that there is the slippery slope, which slippery slopes can be a logical fallacy, but I think in this case it's probably something that we should discuss is, when do you stop? Where is the line? So is it permissive to use gene editing for to help people with ZSD? Well, I think that's probably an easier question to get your hand around. What about, can I use gene editing for height, to make sure that that people are taller than average, or my offspring are taller than average, or that I found a way to cure my male pattern baldness, or whatever, things like that. Is that over the line? Is that impermissible in those cases? And so I think that's a series of questions that we're going to have to tackle before we actually get to the bench. But but having said that, there is the possibility that you would be able to use gene editing. Now, gene editing is probably going to be most efficiently used prior to implantation. So this would be pre-implantation gene editing, i.e., you take a sperm or an egg cell and you do the gene editing on that cell and you implant the embryo. That's probably going to be the lowest bar from a technical perspective, I would think. Again, I'm not a basic scientist. I'm a clinician. So basic scientists would probably be aghast by how overly simplistic I'm making it, but that's at least the way I understand it. I think in terms of gene editing for humans who are postnatal, people who have been born and are currently living, I think that's a more difficult process. Now, gene therapy itself is it overlaps with CRISPR gene editing technologies, but is not the same thing. And so I think you have gene therapies that may use different techniques rather than CRISPR-Cas9, or a different one called TALEN. But still, I think there's maybe some fewer ethical implications for that one. Very long answer to a simple question, but there it is. Other questions from the audience?
Dr. Rush, this is Melissa Bryce Gamble with the GFPD. So I'm wondering if you could connect for our families and the participants today the difference between the retinal gene therapy project that so many of our families are aware of and the CRISPR editing technology. You went into that a little bit, but could you break that down just a bit for our families as well?
Well,
and how that differs?
So I would say that gene therapy is itself not the same thing as the gene editing technology, and so you can use a variety of gene editing technologies, or a variety of technologies, to introduce a healthy gene into some sort of gene therapy vector. And so gene therapy vectors are typically a virus of some sort. And that virus is then you get a healthy gene product that is inserted into the virus, and then you hope that it goes to the right place and changes the genetics such that it either stabilizes a gene, or it halts the progress, or in some cases, if you're lucky, actually can reverse some of the progress. And so in this case, we have Dr. Braverman in conjunction with Dr. Bennett from the University of Pennsylvania are looking at introducing a normal PEX1 gene into the retina of a PEX1 mouse model. So that's pretty. I mean, that's interesting technology for sure. That's the case, and I know that there's a therapy trial underway. Or the therapy trial that's being investigated now by Dr. Braverman's group and other colleagues. But having said the gene therapy itself is just not the same thing as the gene editing technology. That you can do a lot of different sorts of things with CRISPR-Cas9, not just gene therapy. And you can do there's a lot of ways to do gene therapy, not just with CRISPR-Cas9. Hopefully that's sort of an answer. Again, since I'm not a basic scientist, there are some things that I just don't have as much knowledge about than others.
Yeah, thank you. I appreciate that. I think there's this field is becoming so large so quickly that I know many of our families have questions about that. So thank you for helping to break that down. Katie and I are going to see if we have any other questions for Dr. Rush during this time. Thank you all for being here today with us. Katie, I'll pass it on to you.
Yep, and I just want to give everyone a reminder that you can continue to submit your questions in the Q&A area. It's a feature below the player window, and there's also the chat box feature that I think everyone's more familiar with. You can also, if it hits you afterwards, you can always reach out to us. Our website, www.thegfpd.org. Any of the contact us buttons will get you through to us, and we can always follow up on any questions that you may have thought of afterwards. Dr. Rush, thank you for today. I'm going to continue to see if any other questions come through, but I really think this was very helpful to be able to just lay a foundation for everyone for part 2. I know we went a little bit into the different gene therapy, and that's jumping way ahead, but so I appreciate you taking the time to talk about that and separate out some of these different approaches.
Who is going to be talking about gene therapy?
We're going to do that in the fall. But for everyone to know, we're going to have more information coming out. August is going to be part 2, which we're going to go into peroxisomal disorders 101, if you will. And so I really feel like today you've really laid a great foundation to help all of us have the knowledge about the different types of mutations and things like that, and how that works to be able to help everyone not only move forward with their conversations among the rare disease community, but with their own professionals. So thank you so much for explaining all of this to us, taking the time to answer our questions. To our families and everyone who's participating today, thank you for being a part of this learning opportunity. Please be on the lookout for a survey as well. And again, we will be sharing information about the upcoming webinar that's going to be part 2 to this part 1. And I'm just going to double check before we end and make sure that we didn't miss anyone's questions. Looks like a lot of people are thanking you as well, Dr. Rush. We are very appreciative of.
You're most welcome, and I truly appreciate the opportunity to get to talk today.
Thank you. I hope everyone has a great rest of their day.
Perfect.